and anaerobic systems. But up to that stage, people are only thinking that anaerobic systems can produce MFC because of less loss in protons. This is the first report we have published in Environmental Science and Technology, and this have given lead to development of anoxic bioreactors, which can be directly implemented into activated sludge process. And similarly, we optimize the cathode microenvironment also, where aerobic system showed higher electrogenic activity. And we carried out some basic studies on electron transfer. The electron transfer is the major mechanism in which microbial fuel cell is functioning. The electron release from bacterial activity will have to move from aqueous phase to anode. In between there are a number of losses, and when we carry the electron without much losses to anode, it will have a much higher electrogenic activity. So initially we tried to study the function of biofilm in extracellular electron transfer. For that we have used 3 microbial fuel cells. Biofilm with mature biofilm growth with 10% biofilm like coverage, and without biofilm. You can see from this figure significant higher activity was observed in mature biofilm growth, higher electrogenic activity, and we attributed it to direct electron transfer without losing any electrons. And in continuation to that, we tried to evaluate the role of suspension and biofilm on electrogenic activity. And this here, we used Pseudomonas sutida, Bacillus subtilis, a gram-positive and gram-negative strain, which with mixed culture, we augmented mixed culture. After augmentation, the system showed increment in electrogenic activity, and interestingly, we observed that gram-negative bacteria specifically enriched on anode biofilm, whereas gram-positive bacteria enriched in suspension. We are carrying extension to these studies why the specific enrichment was there. And similarly, we tried to enrich the electrochemical active bacteria by giving a small potential before starting up a reactor. And we observed that specific enrichment of proteobacteria was observed after giving potential, which again showed a good electrogenic activity compared to the control operation, which is not given potential. And in continuation that during the process of our research with MFC, we tried to develop different reactor configurations. Initially, we tried to operate dual chamber systems, classic microbial fuel cell systems with anode and cathode chamber. Cathode will be an aerated chamber or a ferricene and cathode. And subsequently, we moved to single chamber MFC where anode chamber is there, which is very much similar to aerobic reactor. And there is a cathode where oxygen from air is taken, and the reduction reaction was there. And subsequently, we have eliminated a membrane and developed a sediment MFC. And so we then came to hybrid system with multi-electrodes, and then multi-chamber MFC, which can accumulate power. Instead of single fuel cell, when we divide the volume of reactor, we can accumulate the power. And afterwards, we developed a pilot plant system, pilot scale MFC with 100-liter capacity, without any proton exchange membrane, and with anoxic microenvironment. And this is giving about 36 milliamperes of power, and which can light the LED lamp of 1.5 volts for almost overnight. This we have used with operated with domestic sewage for almost 1 year, and trying to patent the pilot plant. Specifically, we have designed MFC for solid waste. All these MFCs earlier I have described was for wastewater. This is for solid waste. We try to optimize the electrode assembly, which is showing good results. And when during my first slide, when I talk about MFC, we found that MFC showed a significantly higher wastewater treatment efficiency, and we tried to study why the advantage is there when we use BET, and we named it as a bioelectrochemical treatment system. And our first report was published in 2008 in Biosensors and Bioelectronics, and many people around the world are now looking MFC as a viable treatment area. And here, initially we started working with pharmaceutical wastewater, which is very complex wastewater, and we compared with conventional aerobic treatment and BET, that's MFC treatment. We found almost all polluting parameters are removed efficiently in BET system compared to aerobic treatment. And more interestingly, we observed TDS removal also from BET system, which was not in biological in aerobic treatment, because biological treatment cannot take up the TDS. So along with that, we evaluated toxicity. Compared to aerobic treatment, toxicity levels are significantly lower in BET treatment systems. And we tried to evaluate bioelectrochemical behavior. This cyclic calorimetry of ANT, BET. ANT showed only reduction behavior, but BET showed both reduction and oxidation behavior. The simultaneous reduction and oxidation might be the probable reason for enhanced treatment efficiency. And same thing we applied it for different wastewaters and observed same treatment efficiency. And also for solid waste with petroleum right? Here we observed some interesting observations where the aromatics, complex aromatics can break down into simple aromatics after treating with BET compared to the conventional aerobic treatment. And in order to enhance the wastewater treatment efficiency of BET, we try to regulate the circuit operation and biocathode involvement also, which showed good success. And up to this, we are working on a dark fermentation process. 5 years back, we diverted our research a little bit towards photosynthetic machinery, because photosynthetic machinery has a lot of advantages where it can take both CO2 and carbon from atmosphere as well as water. And people are looking photosynthetic machinery as a viable tool for many value-added products along with biodiesel. So we try to study the wastewater treatment with bacteria, algae, plant as a photosynthetic tools using anoxenic and photosynthetic mechanism, and try to evaluate heterotrophic and autotrophic mechanism to generate biodiesel, hydrogen and bioelectricity. I will start with biodiesel production, where the biodiesel production we try to evaluate heterotrophic mode, mixotrophic mode, where we use wastewater as substrate. We get treated wastewater along with CO2 from atmosphere it will take and convert into oxygen to algal metabolic activity. The cultivated microalgae is harvested and extracted oil and converted by transpiration to biodiesel, which can be directly used as a fuel for transportation fuel. When we compare the properties of biodiesel, microalgae-based biodiesel with fossil-based biodiesel, it's almost as per ASTM standards. All the standards are meeting ASTM standards. In some cases, it's better than the fossil-based biodiesel. And in continuation to that, we try to initially, our major objective is to use mixed culture as a microalgae. For that, we try to extract microalgae samples from different ecological water bodies of Hyderabad and selected about 5 water bodies with different functional activity and taking different kinds of role. And after collecting the mixed microalgae, we transferred to laboratory and operated open pond cultivation in mixotrophic mode using domestic sewage as wastewater, and tried to, and after 14 days of harvesting, we extracted lipid, and that lipids was transfixed, and the biodiesel we found some interesting observations where Nacharam Cheru microalgae we have collected showed higher lipid productivity, about 26% of lipids as compared to the Uppal Pond, Hussain Sagar and Moosi River, because this Nacharam Cheru and Sarura Lake only feeded with domestic sewage, but other systems are fitted with both domestic and industrial effluents, so the diversity of microalgae was not much. And similarly, saturated fatty acids, which is important for biodiesel production, are more in Nacharam Cheru and Sarur Nagar Lake. And we continued with Nacharam Cheru Lake. We designed a mixotrophic mode of operation with 2 phases. The one is growth phase, where we are trying to generate biomass and accumulation of carbohydrates. In second phase, we try to give some stress conditions by regulating nutrients where biodiesel is formed. And by way of this, we are getting about 30% of lipid accumulation, lipid synthesis in dry weight of algal biomass. And among the 30%, 12% was neutral lipids, which is directly responsible for biodiesel production. And we are extending this study with different stress conditions in order to enhance biomass growth as well as lipid productivity. In another, with respect to photosynthesis, we also try to generate power using plant rhizosphere. Normally, plant rhizospheres are rich in organic matter, and when we introduce electrode in the rhizosphere of plant, we can generate energy. This is the basic objective. Because this rhizosphere, because of organic matter, contains a lot of bacteria, which will give electrogenesis. So that we have selected a specific plant which have good root zones and observed good electrogenic activity. Depends on the placement of electrode. Near to root, the electrogenic activity was higher. And we also observed that the photosynthetic activity has direct influence on the electrogenic. Like during night time, where photosynthetic was not there, lesser electrogenic activity was observed. And during daytime, more electrogenic activity was there because of higher photosynthetic activity. And this we are trying to design a system where we can implement by directly introducing electrodes into the plant so that we can generate power in remote areas where power was not there. And similar to that, we have designed based on the plant MFC and the sediment MFC, we designed a system with ecologically designed thing like floating macrophytes based system, which is at present we are operating with heat tanks, which can generate about power of 1.5, light up by power of 1.5 LED lamp. I want to show a video. This is the ES system designed with 3 microphytes, submerged and floating microphytes. This is capable of producing power which can light a clock and LED lamp of 1 millivolts power for almost overnight, 12 hours. The clock is also working as well as LED light of 1.5 volts. Continuously for 12 hours we can operate this system. Before concluding, I would like to say that standalone system, single system cannot work for complete removal of wastewater or complete recovery of energy. So we try to integrate biohydrogen with photobiological process, anoxygenic bioplastic and algal cultivation. And we observed algal cultivation and bioenzymatic process with bioplastic seems to be feasible for upscaling. And apart from basic research, we are also doing some applied research. I will briefly go through it. Like we have designed a semi-pilot plant, full-scale system for plant chemicals, which is at present operating. And we also developed a process for effluent JETL, trigonometric effluent treatment for treatment of ITDS effluent. And we also developed a biofilm system, anaerobic biofilm system for NATCO Pharma. Initially, we operated a lab-scale system and demonstrated at industry with 50 liters capacity, and then applied to DSAR for a semi-pilot plant system. And we also have developed a process by integrating USB and actuator sludge process for common effluent treatment for BDA, by bug drug association. And we have carried out series of projects for ETP performance evaluation for AP pollution control board, Andhra Sugars, and number of EAA studies for Bharat Petroleum Corporation. And I would like to one project which is a challenging project, I would like to briefly explain. This is regarding mercury estimation. AP Pollution Control Board asked IICT to monitor the mercury in contaminated soil near to industry. It's about 20 kilometer stretch river stream. We developed a methodology and estimated collected samples, stream water and soil samples from 20 points, and tried to estimate mercury by ICPMS, and observed that 3 of the water bodies, 3 of the soil samples showed exceedingly higher concentrations of mercury. And then we studied the toxicity leaching test of that mercury soil and observed there is no leaching. So, and again we studied the soil properties, and based on these 3 we have proposed a specific methodology with 4 steps and recommended APPCB to decontaminate the polluting points in the stream. And apart from industrial research, because of our interest, we have done some general research pertaining to environmental issues. This is a PhD program of one of our PhD students, where we tried to do a comprehensive study. The student is from Prakasam district, and we have taken a problem related to that area. It's a severely affected, fluorosis affected area in Andhra Pradesh, and we designed a methodology to estimate fluoride in all the components like water, soil, and biochemical samples and crops. And we observed all the water samples. We have monitored in spite of seasons exceeded about 90% of the samples exceeded the permissible limits, which is prescribed for drinking water, and other parameters. And groundwater showed highly exceeding the fluoride concentration. And calcium pH nitrates, which have direct correlation of fluoride present, showed relatively positive correlation. And so the area, geologic profile of that area is fluoride, fluoropeptide and green granite. The soil is of black cotton, and all the soil samples which we have collected showed the presence of fluoride in it. But pH it's 6.7 to 8.9, which does not leach. And all the dietary materials such as millets, cereals, spice, pulse, vegetables, and other agriculture products showed higher fluoride content than the prescribed 2 mg per day of dietary requirements. And this is because of they are using groundwater or surface water, which is having more fluoride content, which is again going into the crops. And similarly, we try to evaluate serum and urine samples in this affected area, and we observed that all the serum samples and urine samples showed exceedingly higher concentrations of fluoride presence. Like limits are only 0.2 for serum and 1, but all samples exceeded the limits, which is towards skeletal fluorosis. And based on this data, we have suggested some remedial measures to the government authority in the conclusion, by giving conclusion in the chapter. And similar to this study, one of the PhD student, he is a technical officer in IICT dispensary, Dr. Kiran Kumar. He worked on this aspect, like we try to estimate estrogens, which are potential pollutants, and this is the first kind of study reported in India. These estrogens have direct endocrine effect, even at nanogram level, have significant health hazards, like it can cause infertility, obesity, cancer, and many other health hazards, even at nanogram level. And we try to evaluate estradiol, ethyl estradiol. One is a natural estrogen and a synthetic hormone from 7 water bodies in Hyderabad. And we observed almost all water bodies showed alarmingly higher concentrations of both the estrogens. And we tried to, because they are in nanograms, conventional treatment technique can remove this pollutant. So we tried to evaluate 3 of the important treatment methods. One is adsorption, ecological engineering system, and DET. which can tackle the nanogram pollutants and observe that the ecological engineering systems are capable of removing 90% of the pollution of the estrogens. And before concluding, I would like to say a few points about energy and the relation to my work. And when we link these figures, at present people are energy. Everybody knows about the energy requirement and energy demands. And the energy demand has increased, the incremental increase from 1975 to 2010. At present, 12,000 million tons of oil units are being used as energy, and in another 15 years, the energy demand increased by 30%. And at present energy consumption rate, at present we are using mostly depending on fossil fuels for energy demand. At present consumption trends, the existing fossil fuel reserve only there for another 100 years. More precisely, 30% of oil reserve will get depleted in another 30 years, and 70 coal reserves will be there for another 70 years. So by present